Hi, I'm John Quackenbush, and today it's my pleasure to tell you about some of the work I and my colleagues have been doing to try to understand the complexity in cancer, the factors that drive it, and the importance of embracing it. And in the process of doing that, I'm going to talk about networks and network methods, because what we've seen is that networks are an extremely effective way of capturing and dealing with the complex interactions that we know drive the development and progression of many diseases, including cancer. So I'm going to begin by really framing this in the context of the human genome. The genome, as you know, is more than 22 years old. It's been announced finished many times, including with the completion of the Y chromosome earlier this year. But even 22 years ago, when the genome was first announced finished, we had a collection of 25,000 genes. And people started to think that these 25,000 genes were really a parts list for making a human cell. And that by understanding variations that occurred in the genes, one could then understand the factors that contributed to disease development and progression. And it's a great idea, but it doesn't really capture the full complexity of biological systems. And the easiest way to think about that is simply to think about your body with more than 250 cell types, all of which have essentially the same genome, each of which has a different morphology, carries out different functions, behaves in a very different way from the other cells in your body. So it can't be just the genes, although we know that the genes do play a role in determining our phenotype, but it must be something about the control or regulation of those genes and the way those genes and gene products interact with each other that make cell types different, that make you me, but then also distinguish between health and disease. So how do we figure out what those genes do and how to work with them? Well, there have been a lot of ideas over the years about ways in which this problem could be tackled. One of them was using AI and machine learning tools. And granted, a few years ago, they weren't nearly as sophisticated as today. But these efforts really failed in being able to capture and deal with the biological complexity that's so important in the kind of analyses we like to do. And one example of that is IBM Watson, which, although it succeeded at many other tasks and won on Jeopardy, it really wasn't able to ingest all of the complex, sometimes contradictory biomedical literature and to be able to match individual patients with their genetic background to individual diseases. So, why has AI struggled? Well, there was an interesting paper that appeared in the MIT Technology Review now just about 3 years ago that was entitled The Way We Train AI is Fundamentally Flawed. And they listed two independent problems. One is the problem of data shift. So if I train an algorithm here in Boston and then attempt to apply it in New York or San Francisco or London or New Delhi, it may fail for a variety of reasons, one of which is the fact that the way in which we encode data here may be different than the way in which data is encoded at these other institutions. There may be racial and ethnic differences in the population that influence what we see. There can be a whole host of different factors based on the shifts in data itself that cause the algorithms to fail, to potentially learn artifacts rather than learning the true drivers of conditions. The other problem is one referred to as underspecification. And this is the problem which is well known in statistics, where observed effects can have many possible causes. And often, without a complete understanding of the systems we're trying to model using AI and machine learning, we may fail to capture those interactions. And so we may have an incomplete picture, which then leads to improper conclusions. So I really think about this in terms of two basic shortcomings. One is insufficient training data, which means we need more data from more populations, as standardized as it can be, but really that captures the complexity of what happens in the real world. But the other piece that's important is building a robust underlying model. And this is something which isn't new. One of my favorite papers in all of computer science is the 1997 Wolpert-McCready paper on no free lunch theorems for optimization. And if you read the first sentence in the introduction, it really sounds like an indictment of almost every single machine learning paper I've read for the last 10 or 15 years, right? The past few decades have seen an increased interest in general purpose black box optimization algorithms that exploit limited knowledge concerning the optimization problem on which they're run, right? We take a big machine learning algorithm, we take that linear algebra machine, we feed in all of our data, and truth pops out. And that's not really a practical way to employ these systems. In this paper, what they do is they go through a series of arguments and analyses. And what they show is that in these large-scale optimization problems, which is what machine learning really is, that with realistic data sets in the amount of time we have and the compute power available, that the best way to get arbitrarily close to a near optimal solution in finite time is to exploit knowledge about the system that we run, right? These same results also indicate the importance of incorporating problem-specific knowledge into the behavior of the algorithm. So this is something we should be in a position to do. And one of the things I'm going to argue today is that one way of introducing those constraints is to use networks. So a network is really a sort of an abstract idea. It's a way of representing complex association patterns between various entities. And the nice thing is I can draw a network as a graph, and I can represent that graph using what we refer to as an adjacency matrix, right? A matrix that shows connections between the nodes. So if you look down here at the bottom, I have 3 different nodes numbered 1, 2, and 5. And you can imagine these being various elements of the system whose interactions I want to try to record. And then I have edges, and the edges connect 1 to itself, 1 to 2, and 1 to 5. And if you look at the first row, there's a 1 in column 1 and 2 in column 5. And what that adjacency matrix does is reflect the patterns of interaction that are seen in my network. Now, a network can represent a real biological network or physical network in which objects actually interact. But those edges, those interactions can be abstract associations like correlations and patterns of gene expression or other phenomena that we want to try to represent in a graphical fashion. So networks have been incredibly useful for me in my work in thinking about the way in which genes interact to help define phenotype. The sort of standard paradigm we've operated under for more than 20 years involving gene expression data has involved collecting expression data on different phenotypes, doing a statistical test to identify differentially expressed genes, and then trying to interpret those in a variety of different ways. Sometimes people projected these back on networks like protein-protein interaction networks and discussed them as regulatory networks. But in many cases, it's an incomplete projection because it ignores one of the fundamental truths that underlie our analyses, that the phenotypes differ because they're genomic differences. So what we've started to think about is that rather than to have one canonical network that captures the changes in expression, we may in fact have different networks representing different phenotypes. And if we make that leap, what we can then start thinking about doing is the fact that these networks have unique structures and topologies, they differ between the different phenotypes, and that in fact, when we look at the differences in topologies and differences in expression, and look at functional changes in the genes that are regulated, what we can start to do is to tease out additional data and information about the drivers of the system. And eventually, if you carry this far enough, you get to the idea that individual networks themselves may be good biomarkers or descriptors for the individual systems we're trying to study. So my colleagues and I have developed a whole suite of tools that we name after animals for inferring networks. The collection of animals is housed in the network zoo. The URL is down on the bottom, and Mero and Ben Kabila and Tian Wang really did a lot of work in bringing all of these tools that various individuals that I've worked with over the years have contributed to developing. We've also developed a set of online tutorials that we call netbooks. And what they do is they take you through each of the individual tools, step you through their applications, show example data sets. And in fact, netbooks increasingly is capturing every paper or nearly every paper that I and my group write. So these are freely available tools, and I'm going to describe them in a little bit of detail in the time I have today. But the core idea behind them is that biological systems are driven by complex networks. The structure of the network is important and informs our understanding of the system we're studying, and that there's no single right network. The network in each tissue, in each biological state, or in each individual, in you and me, is going to differ. And those differences are important. So people will often ask me, how do we know the network is right? And my answer is always, it's not. It's a model. The real question is, does the model inform our understanding of the biological systems that we're studying? And I'll show you some evidence today that, in fact, it does to a better extent than many of the other tools we've been using for quite some time. So we started this journey now almost 10 years ago, maybe more than 10 years ago, asking a simple question: can we model the gene regulatory process? And this produced one of the first animals in our zoo. It's a method that we call PANDA. My colleague Kimberly Glass really drove the development of this method and has been intimately involved in many incarnations of methods that build on it. And it's based on a simple idea that what we want to do is look at the data we can collect, which is gene expression data, RNA transcripts that we can measure through microarrays years ago, now RNA sequencing. And if we look at those RNAs are produced because an RNA polymerase bound to the genome somewhere. And that was typically facilitated by transcription factors, proteins that bind to the regulatory region of the gene and activate or repress its expression. So what we start thinking about doing is modeling a system in which transcription factors can bind to the genome. And the good thing is we know where they're likely to bind based on data that we have about transcription factor binding sites. So we can look at a transcription factor and its possible targets. But if we have expression data, we can actually refine our guess as to which transcription factors might be regulating which genes. And we can refine our knowledge of the system by saying that if these two genes are co-expressed, are correlated in their patterns of expression, but the third one isn't, then it's most likely going to be true that these first two are regulated by this transcription factor, but the third gene may be regulated by something else. So that's one piece. Another piece of the puzzle that we use to try to refine our models is just the idea that proteins can interact with each other. And in particular, transcription factor proteins can interact with other transcription factor proteins to form complexes. So the idea is that if we have evidence from mapping transcription factors back to the genome, that two transcription factors may interact with each other and they both have binding sites upstream in the gene, that in fact that may be some evidence that these two transcription factors are cooperating in the regulation of this gene. So we can do this kind of analysis for every transcription factor in the genome and every gene in the genome and use it as additional information to fine-tune our models, right? We start with a guess like the no free lunch theorem tells us, and we make our models better. So how do we actually do this in practice? Well, I've alluded to the fact that we start with motif data and we guess a structure for the regulatory network, transcription factors regulating genes. We take the protein-protein interaction data, and we take the gene expression data and look for correlations. And then we take all these three pieces of data and we iteratively refine our models until they converge on a workable solution. So we start with this motif scan of the genome, guess a network, take the other data, and then update the network and our other data repeatedly until we get a final network which doesn't change. And this has turned out to be an incredibly useful paradigm captured in this PANDA method that we've used in a whole host of papers, including papers looking at cancers, looking at environmental perturbations, and looking at how drugs alter the structure of regulatory processes in cells. And we have a lot more applications. There are a lot more papers that we and others have written that have really demonstrated the utility of an approach like this. One of my favorites is just an analysis we did looking at 38 individual human tissues using data from a large-scale project called GTEx to simply ask, how do differences in regulatory patterns change between different tissue types? And it was really interesting. What we saw was there are core regulators and regulatory processes that were more or less unchanged between different cell types. There was sort of an outer layer of stuff that was changing, but the really interesting and tissue-specific changes occurred sort of at a middle level, where what was happening is transcription factors were adding or losing different genes they were regulating. And as a result, the regulatory processes were changing and altering tissue-specific patterns of expression. So this was really exciting because it starts to answer one of those first sort of questions I put up. How does the same genome produce different cellular phenotypes? And the answer is tissue-specific patterns of regulation. But I also started with this proposition that we want to move beyond a single network. And our attempts to do this really grew out of some analysis that a postbac student, Matt Tuggen, did. It got Kimby and Marike Kujer in particular very excited. And what, in talking about this problem, we started to recognize was that the network that we estimate from a population is really an aggregate network capturing the effects of all the individuals. But what we should be able to do is to tease out those individual samples. So we developed an approach based on linear interpolation that we call Linus. And the way Linus works is pretty simple. If I look at everyone who's listening to this presentation now and collected blood samples, I could generate a network for the attendees of this presentation. But that network is going to average across all of us. If I then leave you out and estimate a network, if you were perfectly average, leaving you out wouldn't change that network at all. If you had one connection that was far stronger than everybody else's, then leaving you out would reduce the estimate for that connection. If you had one that was far weaker, leaving you out would increase the estimate for that edge. And so if I look at the network with you and the network without you and subtract them, what I get is an estimate of the differences between you and the average network. And if I'm really averaging, I can take that average difference, I can scale it by the number of samples, and I can add it back to the network without you to get an estimate of your network. And so I now put you back in the population, I take me out, and I do this for everybody listening to this presentation. So now instead of one network for our group, what I have are hundreds or thousands of networks for a group. And now I can take these inferred measures on a population, our population, and I can begin to do analyses of those, treating them as measurements and doing statistics. So we've applied this in a variety of different ways. The paper describing Linus appeared in iScience about 3 years after we first developed it. But we've applied it to a number of really interesting problems. Again, analyzing data from GTEx. We looked at 29 different tissues and inferred tissue-specific regulatory, include tissue and sex-specific regulatory networks to ask how do male networks differ from female networks. We took this further when Camila Lopez Ramos did an analysis of colon cancer and looked at the differences between regulatory networks in males and females. And to her surprise, discovered that those networks differ in the way in which they regulate processes like drug transport and drug metabolism. That got us very exciting because the networks we built were with surgical samples before anyone had been given chemotherapy. Based on those analyses, we would predict that males and females are likely to respond differently to chemotherapy. And clinically, we know that they do. So this information has really been an important way of being able to tease apart the regulatory processes that distinguish clinically relevant differences in male and female disease. We've also applied it in other situations. Marike Kujer and Camila Lopez Ramos, who I mentioned earlier, looked at glioblastoma and, to their surprise, found in a tissue that's supposed to be protected by the blood-brain barrier, that in that tissue, there are actually differences in the regulation of immune processes that were predictive of outcome in the disease. So the question really is, does this approach tell us something that expression or co-expression doesn't? And to convince you that's true, in fact, my colleague Des Weghill and a number of others and I did an analysis looking at gene targeting in disease networks. We showed that you can have situations in which you have no differential expression, but differential co-expression. And remember, co-expression is one of the inputs to our model. And in fact, there are many analyses that people do that are based on creating co-expression networks. So what we were able to do is then look at an example, pancreatic cancer. In fact, Des got all the data from the TCGA. We used Panda and Linus to build individual sample-specific models for every single individual in the population. and then classify them as to whether they had basal or classical subtypes of cancer, and compare them to find differences in the regulatory patterns, and then ask what genes were differentially regulated. It's sort of interesting because if you look at this, the differential expression data is shown in blue. Differential expression found a number of things that no other method found, but those differentially expressed genes really didn't help explain anything about the differences between the two different disease types. Differential co-expression found things that almost completely overlapped with our regulatory network models. But in fact, and a lot of these are relevant to disease, but in fact, many of the most interesting processes, including immune-related processes, epigenetic modification, and cell cycle processes, were only found by looking at gene regulatory networks and differences in transcription factor targeting. Right? So what this is really driving home is the idea that the regulatory networks do in fact capture something which is distinct from expression or co-expression by itself. So we started with the genome and individual genetic differences. So now you can start to ask, do our genetic differences actually interact with these transcription factors to make the regulatory processes unique to each of us? And the answer, of course, should be yes. The question then is how? So Des Weghill, John Platek, and others and I worked on a method which is now called EGRIT. And what EGRIT does is it really takes into account that this process of transcription factor binding itself is complex. So in our original model, we had transcription factors with genes, and we used that to create a preliminary model. But now you might imagine that a SNP appearing in a transcription factor motif may change or alter the binding affinity for a particular edge. And so what we'd like to be able to do is to account for those differences. So we start with a basic panda model that I showed you earlier, but now on an individual specific basis, we model that motif prior. And what it allows us to do then is to build a network for each person in the population that reflects that person's own unique genotype. So we're building this, we're testing on data from ENCODE, where we have genetic information, we have transcription factor binding data, and it was all very encouraging. But where we got really excited about this was an analysis of data generated on 119 Yoruba individuals who had all been genotyped. The Yoruba are a West African ethnic group, very ethnically diverse, genetically diverse. And from those individuals, they created blood and created lymphoblast cell lines. They then created from those lymphoblasts induced pluripotent stem cells and differentiated some of those to form cardiomyocytes. And they collected expression data on each one of these samples. So we have 119 genotyped individuals, 357 gene expression profiles representing 3 different tissue types. We built our models, and we then asked for transcription factors that are associated with different diseases, where do we see disruption of those? And we started with 2 examples. One is coronary artery disease, and we looked for transcription factors that have been linked to coronary artery disease. And what was exciting was we found some differences in the lymphoblast cell lines, some differences in the induced pluripotent stem cells, but the biggest differences in binding potential that we saw were actually those that occurred in the cardiomyocytes, right? Heart disease disrupts regulation in heart cells. But even more interesting was an example of Crohn's disease, right? Irritable bowel syndrome, an immune-mediated process in which we saw no disruption of the associated transcription factors in our model in the heart cells, basically none in the induced pluripotent stem cells, but substantial individual-to-individual variation in transcription factor binding in the cell type that we know is associated with this disease, right? And what it's telling us is that our own individual genotypes influence the way our networks are put together in disease-specific manners, right? So this is exciting because this problem of teasing out various influences is now starting to fall into place. So the final question I'll try to address today is, can we use these networks to help find drug candidates? And again, the answer is yes. Maro and Ben Gabilla, who I mentioned before, worked to assemble now well over 200,000 gene regulatory network models that were either built in various studies we did or as a specific analyses that he did to try to pull together data from the connectivity map and clue. And what he did was he built these into a data resource. And instead of asking what drugs alter patterns of gene expression, Marouin developed a tool called ClueReg to ask the question, how do different drugs or compounds alter regulatory processes? And by using those, what one can then do is say, how do we use a drug to change one regulatory network into another? So I'll just give you one really quick example. In looking at lung cancer, what Marouin discovered was that there was a drug that appeared to alter the regulatory patterns of that appeared in lung cells. But that drug had only been tested as an aurora kinase A inhibitor in colon cancer cells. But what our model predicted was that these regulatory changes would in fact be produced in the same way in lung cells. And it suggested that this drug may in fact be a good therapeutic candidate for treating lung cancer. So we can start to tease out approaches that will allow us to repurpose drugs and change their applications or change their clinical use, really by looking at not just alterations of expression, but alterations in networks. So I know this has been a really high-level overview. I hope I've convinced you that networks are useful, because in various applications, we found that networks provide insight into disease that's not found using expression or co-expression alone. Differential targeting captures not only a phenotype's current expression, but also its potential to respond. And changes in network structure can help identify drivers of disease and, as Marouin showed, potential therapeutic targets. So there are lots of things I haven't told you about. We have methods for ordering samples in pseudotime. You can imagine if we take all of our networks and look at us along the progression from health to disease, we're going to fall at different time points. And by looking at that, we can look at the temporal or pseudotemporal evolution of regulatory processes. We've also developed Phoenix. This is work of one of my graduate students, Inta Hossain, a method that uses neural or ordinary differential equation solvers to actually create quantitative models of gene expression as differential equations that we can model genome-wide regulatory networks over time. We've also been looking at epigenetic age and epigenetic age acceleration as a way of ordering samples, but also to think about the way in which disease can represent altered processes of aging. And then last but not least, one of the things we're about to submit a paper describing is the loss of Y in male tumors. And we have good data showing this is selected for by immune evasion and that it may be predictive of immuno-oncology response. So that's all I have for you today. I'm afraid I'm out of time. I like to use quotes. So this is one of my favorite quotes I like to use in many of my talks. Before I came here, I was confused about this subject, but after listening to your lecture, I am still confused, but at a higher level. So I hope I haven't left you too confused. I hope you enjoyed the presentation. If you have questions, please feel free to either ask them today or to reach out. I'm always happy to respond, and you can tell I enjoy talking about my work. So thank you to the organizers for having us. Thank you all to all the wonderful people I work with, and enjoy the rest of this absolutely fabulous conference.